In this lecture, we'll be talking a little bit about, at the beginning of the lecture, microarrays, which is a fairly new technology used to quantify DNA in large quantities of it and compare samples. And one of the things it's used for is large-scale genomics, and that's kind of what we'll talk about in the second part of the lecture. And we're really not going to get into a whole lot of details here in this class. You can learn a lot more about this if you go into like grad school or medicine. But our level of dealing with this is going to be quite small. All right. So first thing I wanted to mention, and this is probably will make more sense at the end of the quarter, but how do we get to a gene expressed into a protein? You start with genomic DNA. So this is our genomic DNA. And as we've sort of alluded to, there are different pieces in the gene. Some of the gene is what's shown here in red is an intron, and those don't get converted into proteins. So as the DNA gets converted to RNA, these red pieces that are introns get spliced out, and that's eventually what turns into our mRNA, messenger RNA. That goes on to be translated into proteins. Okay, so I just wanted to point that out because that will probably come into play here. And in some of these microarray techniques, we're going to be talking about using RNA and cDNA, and so this basic idea of a gene gets transcribed into RNA, part of the gene is cut out to make messenger RNA, that gets translated, will be important to know. And again, that's going to be, we're going to cover this in detail, the process which this happens in chapters 25, 26, and 27. And that'll be at the end of the quarter. Okay. But right now, I think it's good to have a picture of it in our heads here. Okay, so that brings us to microarrays. So with these, generally you're asking how much of a gene is expressed. In other words, what messenger RNA sequences are present in a particular sample. So how these chips are made are shown here. This is probably not going to be super important for our class, but if you're interested, you have a solid surface, and then on that surface you put, in each spot, you put a sequence that would represent a different gene or a different part of a gene. Right? And how they do that is they use, first, they introduce a nucleotide that they want to start the sequence with, and they polymerize it to the chip. That would be the solid surface, is like a microchip. They polymerize it in these specific spots they want it to by reacting it with light. Right? And they just cover the spots they don't want that particular nucleotide to be. So in this example, they're putting an A down, hit it with light. It's on these two spots. Then they add G, hit that with light. It's on these two spots because these two were covered, and so forth. So you can make unique sequences at all these different parts on the chip. Okay. What this is going to be used for primarily is gene expression. So you can say, is this gene expressed in this tissue? So you can take a particular tissue and isolate your DNA or messenger RNA from that, and then put it on the chip and then read it. And we'll see how this is done here in a second. Okay. The other point that it's really used for a lot is in development. So certain genes are expressed at certain times in an organism's lifespan. So that understanding when a gene is on and off is important to some researchers. So that's another way these are commonly used. Right. The photolithography, that's the process in which they make the plates. Okay. Right, so how do we use them? So this is an example of a typical way a researcher might use this. So you could isolate your messenger RNA from an organism here that's at two different states of development. Okay. Right, so they isolate messenger RNA from that. And then what you do with the messenger RNA is, fairly unstable and harder to work with. So you use reverse transcription and you convert that back into DNA, and we call that DNA cDNA. Okay? And that's the cDNA is complementary DNA, and it's just a measure of what DNA is actually being converted into your messenger RNA, which gets transcribed into your protein products. Okay, so it's your genes without the introns. Right? And when you do this, you label, one with a particular color, let's say green here for that egg, and then for the full-size organism there, we use red. Okay, so you use two different colors so you can tell them apart. Right? You mix them together and then you apply them to your DNA microarray chip. Okay? And those sequences are going to hybridize on the dots that they match up with the sequence. Okay? So if your sequence is there, you're going to see a fluorescent color when you read these. So the spots that have cDNA from the egg cell, they're going to show up green. Then they only have the DNA from the egg cell. The spots that have DNA that's only from the mature organism are going to show up as red. And then when you have a mixture, you're going to show up as kind of a mixture of yellow and red, green and red, which would be yellow. Okay. Right, and so here's an example of what a real one might look like. Okay? And so you would use a computer and analyze all these spots, right? All these spots, you would know what sequence is there, and you can kind of correlate that to gene expression. What genes are expressed more in the egg cell, what genes are expressed more in the mature organism, and the combination thereof between the two, if they're mixed, if the gene's expressed in both cells. Right, so that's microarrays in a nutshell. Another medical use of these is in cancer. So the expression of genes varies a lot between tumor to tumor. And so if you use a DNA chip like this, you can get sort of what's known as a fingerprint of the tumor to have an understanding of what genes might be overexpressed in the tumor cell, and those are often targets for treatments. Okay, and so you can correlate that with how certain drugs work and determine the best course of treatment. Okay, so we're going to move on to the section 9.3, and that's the last part of the chapter 9. This is really going to be a short lecture. This stuff isn't going to show up as much on the exam as the other material, but I think it's still interesting and good to know. Okay. So we're going to talk first about some methods of DNA sequencing, and I think the book was rearranged a little bit in this newest edition, so some of this material might have been covered in chapter 8 in the book, but we're going to discuss it here now. I think it's still relevant. Right. So there's been a new generation of DNA sequencing, and it's referred to as next-gen sequencing, and it employs microarrays. So the full genome is immobilized on chip, and it's broken up. The genome's broken up into fragments. So at each spot there's a different piece of the genome, right? And they're about a few hundred bases long. And then you can do these sequences of all these little pieces all at once, and that allows faster detection. The first human genome that was sequenced took years to complete because you're doing it one at a time, kind of in a sequence. This you can do many bases at the same time and get it done much quicker. So this can be used in a clinical setting now, whereas 20 years ago that would really not have been the case. There's different methods for doing this. There's quite a few actually. We're only going to talk about two of them. One is pyrosequencing. Okay? And so for pyrosequencing, it involves synthesizing DNA from a template. Single nucleotides are added at a time. Each nucleotide added results in a pulse of light. Right? So that's sort of the pyrosequencing. It's kind of like a burst of light that you're reading. And with this, you're reading about 400 to 500 nucleotides in a sequence. Right? So how this happens. So your chip DNA, we'll just say is this piece here. And then you add your primer, and then you pulse in different nucleotides at a given time. Right? So here you pulse in A. A isn't matching up to the next nucleotide in that strand, so nothing happens, no flash of light. You pulse in a G, that matches up here with the C, then there's going to be a pulse of light when that is added. And it comes into this reaction. It uses the nucleotide triphosphate to go to nucleotide diphosphate and inorganic phosphate, right? And then the nucleotide monophosphate to paraphosphate, this transition here done by another enzyme is what creates the light. Okay. And that reaction is just in general is shown. Okay. And so what you're reading would look something like this. You're adding A, you get a pulse of light. You add G, you get two pulses, or a double size pulse of light, right? Double intensity. That means two G's are back to back in that sequence. Then T, then C, then you add A, no pulse, there was no A there. So that's kind of how you're reading what the sequence is. Right, and how it looks on the chip is shown here. So at all these different spots, you can tell, right, is A in that sequence, in this circle, yes, in that circle, no, there's no A in that particular time, okay? So each different cluster is a different sequence, and you can read them all simultaneously, so that speeds up the process here. Right. The enzyme that's commonly used in this, luciferin and luciferase, right? So you have this chemical luciferin that's converted into this form that gives off the flash of light, and that's done by luciferase, which is first isolated from fireflies. Okay. Right. The next method we'll talk about in next-gen sequencing is what's known as reversible terminator sequencing. It's going to look somewhat similar to pyrosequencing. Right. The difference here is that you're not using this chemical reaction to generate light. You're using fluorescently labeled nucleotides. Okay. And so in some ways this makes it a little bit easier to understand, or less, probably less complicated. So here we have our sequence that we're hybridizing to or trying to read. That's on the chip. We have a primer. We add nucleotides, and you can add them. You add them all at once together. That's the difference here. You're not adding one at a time. You're adding them all together. When it pairs, they're what's known as blocked, so that there's no fluorescence until they're added into that incorporating strand there, right? So you add all four of them. The one that gets added is the one that's going to be unblocked and give off its particular color of fluorescence. So here, the T was the next nucleotide, gets added, gives off a yellow color, right? Then you kind of flush the system, remove those, you add them back in, and the next one here that gets added is an A. It gives off blue. Okay, you flush them away again, add some more, and you keep doing this. So in a similar manner, you can do this on a microarray, many different spots at the same time, and the data that you're getting is all just in colors. So based on what color is given off, that's the nucleotide. Right. So doing it in this microarray sort of manner it really sped up the process of looking at, you know, the human genome and in genomes of all organisms actually. Right. And so some surprises have come from this after researchers have looked at enough of these genomes. Right. When the first human genome was read, you know, it was just one individual. But after getting many different genomes and comparing them, you can kind of make a better comparison and learn more. Right? So one of the big surprises was that we have only about 25,000 genes, as opposed to 100,000, which was more of the estimate. Okay, so a much smaller number of genes than was kind of predicted. Right? And that's only about twice as much as a worm has. So that's kind of crazy to think, you know, we think of ourselves as being much more complex than a worm, but we only have about twice as many genes. Okay. We've also noticed that we do share genes with many organisms. And this is one of the lines of evidence to support evolution. Right, because you have a lineage of genes that you can trace throughout lower to higher organisms. We don't really know what the purpose of much of our DNA is. So that's kind of one of the crazy things. We think of, in this class anyways, we think of DNA being sort of a recipe or an instruction manual on how to make a protein, right? A gene encodes a RNA, that encodes a protein. But only about a 1.5% of our DNA bases encode for protein. So that's a very small amount of it. So there's much more DNA that's used for other things, and a lot of that usage we really don't know what it's for. So looking through the chapter, or this section anyways, I found some important terms, and I think this is probably the most important thing to have some understanding on from section 9.3. Okay. And we'll see some of these terms again, right? Introns, talked about that at the beginning of the lecture, but those are sequences of a gene that get spliced out before that messenger RNA is translated into protein, right? So introns is kind of short for intervening sequences. Okay. The opposite of that is an exon. An exon is a sequence of a gene that does encode for the protein, that does make it into the messenger RNA. Transposons, that's kind of a short term for transposable elements. Those are sequences within a genome that can move around or jump around, right? They transpose, they can move. And we'll see on the next slide that a lot of the human DNA is in the form of transposons, transposable elements. I don't personally know a whole lot about transposons. Looking just briefly about it, it seems that at least for human DNA, the source I found said that none of the transposons in human genome are currently active, and they're thought of as fossils. But they're at least if you look at the amount of DNA we have, a good portion of it is from transposons. You know, whether or not they're actively moving around, uh, you know, notwithstanding, there's still a lot of DNA from that. Simple sequence repeats, SSRs. Okay, these are generally less than 10 base pairs. They're repeated many times, right? They could be repeated, you know, thousands of times, even millions of times. What structures in the chromosome that these make up, and you know, we'll see this in chapter 24 a little bit, but the centromeres and the telomeres are made up of these simple sequence repeats. Okay, so and we'll look at we'll actually see some of those sequences in that chapter. Another term you might see is short tandem repeats, STRs. Those are used in forensic DNA analysis. So, you know, every individual's has this kind of unique profile of short tandem repeats that you can use to identify people via their DNA. So that's kind of what that's used, can be used for clinically or forensically. Right, and then the final term that the book kind of brings up and highlights there is what's known as a single nucleotide polymorphism, or these are also referred to as SNPs. Okay. What SNPs are is, you know, it's just in your sequence of DNA, you have one nucleotide that's different. Okay, that would be a single nucleotide polymorphism. Okay. And there's a natural variation between individuals in these. Okay, so typically it's something like 1 in 1,000 base pairs. You're going to have just randomly different nucleotides at specific positions. And they don't, you know, classically, you know, if you have a change in a nucleotide, you could be changing an amino acid, which would be, you know, causing a mutation in the protein, which could be giving you some sort of a negative effect, right? But this doesn't really include that, right? This is a nucleotide that's changed, and it might not even change the amino acid it encodes for, right? It does, and it's not giving a negative effect at all. Okay, so they're similar but different than a point mutation. Okay. One of the things that these are used for, these SNPs, you can use them to determine what's known as a haplotype, and that's used in genealogy, and you know, websites like 23andMe kind of use that to kind of show your ancestry. And so people with you know, from a particular ancestry will have similar single nucleotide polymorphisms. Okay, that's one of the ways they can determine haplotypes. Right. I should mention too with the SNPs, SNPs can kind of make discovering mutations in patients a little bit more difficult. Say you have a patient that's in the clinic, you don't really know what disease they have, and this happens at Anschutz and in the clinical side of our section quite a bit, is where there's a kid that very young that's presenting with some phenotype that's not well characterized. They don't know what the exact problem is, so they have to do some searching. And typically you'll have an idea of what kind of pathways are involved in this, so you know what genes to look at. But just the sequencing of these genes, it can be tricky because you'll find what you think is a mutation possibly, but it can be a benign mutation, and something like a SNP can show up. Okay. So this figure is a little bit different in the current edition. It kind of classifies these transposons a little bit more. I think there's one extra section here, not super important, right? For our class, I really want you to focus on protein coding genes. That's a 1.5% of our DNA. Introns, this is almost a quarter of our DNA is intronic DNA, which is a very large amount. Then you have the repetitive sequences, the longer repetitive sequences like centromeres and telomeres, about 8%. Simple sequence repeats are about 3%. There are some duplications that have happened, that's about 5%. And just miscellaneous unique sequences are 11.6%. So this stuff here is probably the more important stuff to know, and in particular, protein coding, 1.5%. That's probably what you really need to know. And the takeaway too is that a lot of our genes are intronic and get spliced out before they're made into proteins. Right. So genetic testing, now very cheap. You can do this stuff like 23andMe. There's other ones. It's like $100 or something, and you can get your whole genome kind of done. It brings up ethical issues. You know, if you know you're going to predispose to certain conditions, right? It just brings in ethical questions, right? Like, okay, should you really, have children with this person that has similar defects in their genome, and you could pass that on to, the next generation. It makes human disease, especially cancer. Cancer is a very complex disease. On average, it's something like 6 to 8 mutations have to happen inside a cell before it becomes cancerous. And so there's not just one cancer, right? It can be very, very complex. But having this data with whole genome testing on, millions and millions of people now brings this into focus where you can actually get a better idea of how to treat particular cancers and how to get a genome, let's say, on a particular cancer cell and make much more targeted treatments. Okay. Health insurance cannot discriminate based on genetics, or at least it can't currently. So that's one thing that's good. But in the future, who's to say that it might be allowed, right? So that brings up more ethical questions. Right, there's this video here by Richard Resnick, this link. It's from a TED Talk. It's 11 minutes. It kind of delves into these questions, and it's an interesting watch if you're interested, right? And so one of the things that I've done is I've done 23andMe, right? I'm not going to say most people, but a lot of people have done these things. Another interesting site that I was kind of told about by a grad student in our lab at Anschutz was this Prometheus, and I haven't checked this recently, but what it was a place you could take the data from 23andMe, upload it into this site, and it could give you kind of a idea if you're going to be more or less likely to have certain diseases or disorders in later in life. So it was one of those things that do you really want to know that stuff, or is ignorance bliss? But I thought it was pretty interesting, right? It kind of went just a step further than the the analysis they gave you on 23andMe. Right, so that's all we really wanted to discuss here about microarrays and genomics. The next part of the class will be moving into discussion about genes and chromosomes. So that's chapter 24.